This is part 2 of our discussion of chapter 2 of our exercise physiology test. In part 1, we introduced the idea of homeostasis, and in addition we said that during exercise, the quote normal range of variables, the conditions in your body, may be shifted to consistently new values called steady state values. Now we're going to discover that in addition, challenges to your body during exercise can lead to more permanent changes in the entire homeostatic mechanism. So, during exercise, there are pH changes, drops in oxygen pressure or concentration in the body fluids and the cells, increase in carbon dioxide pressures or concentrations, increase in temperature, and usually our biological control systems can again maintain those within some steady state ranges during exercise, at least in a cool environment. There are conditions in which the environment is too hot to dissipate all the heat produced during exercise from the body, and if there's too much humidity, evaporation can't take place, and so we may exceed the body's ability to maintain a steady state, and the body temperature, for example, may continue to increase. The pH of the blood may continue to decrease. The result is fatigue and cessation of exercise. We talked about the control systems that maintain variables, including a receptor, a control center, and effectors that can reduce the stimulus, the change in variable that occurs during exercise or during normal regulation. If any one of those components fails, then homeostasis cannot be maintained. We call that a disease state. So an example of that we could introduce is diabetes mellitus, a familiar disease in our culture. Type 1 diabetes mellitus involves death of hormone-producing beta cells in the pancreas. An autoimmune attack is thought to be the basis for killing those cells. So then insulin is no longer produced by the pancreas, and that means that cells of your body can't take up the glucose, and the result is hyperglycemia, an increase in blood glucose levels. So the dietary glucose that you consume in your carbohydrates just remains in the blood. So in this case, we're talking about a failure of the receptor and the control center. Both of those functions reside in the beta cells themselves. They monitor blood glucose levels, and they in turn integrate and release the hormone insulin in order to act on the effectors, the cells of your body. Type 2 diabetes occurs with increasing fractions of adipose tissue. Obesity is associated with insulin resistance. Insulin is released by the pancreas, but the cells of your body can't respond to it, so they can't take up glucose, and again, the result is hyperglycemia. In this case, we're talking about a failure of the effector cells. The effector, I should say, the cells that normally take up the glucose. There's still insulin there, there's still a receptor and so forth, but the effector can't remove the glucose from the blood. Adaptation refers to changes made in your body in response to challenges so that we don't just react the same way every time to particular challenges. Challenges that the body meets with on a regular basis lead to relatively permanent changes in the way the body responds to those challenges. So homeostasis, the whole process, may be changed in order to better react to a particular type of a challenge. It's called adaptation. In a particular instance of a environmental stress, we call it acclimation, or some people call it acclimatization. Some authors try to distinguish between these two with respect to the duration of the time frame, calling acclimation a shorter term change and acclimatization longer, but we won't worry about that too much. But we're talking about adaptation, changes in your body in response to environmental stresses. In other words, for example, if you exercise in a very hot environment, you can become better at exercising, better at maintaining your homeostasis and your steady state conditions in your body in the presence of a hot environment as you are exposed to it more and more. Oh, high altitude is another good one. If you go up to high altitude, there's less oxygen in the atmosphere, and initially that produces quite a uncomfortable experience. Altitude sickness is called, but after a relatively short time, your body adapts. It changes the way that it manages its ventilation, its breathing, and also it changes the amount of hemoglobin in the blood, and voila, you can operate comfortably at a lower oxygen content in the atmosphere. So that's called acclimation, or it's a form of adaptation. Hormesis is a term used to describe those kinds of things. Hormesis describes changes that your body makes to improve its ability to respond to stresses and to maintain homeostasis. The thing about it is there's an optimal challenge that will induce hormesis, and there are challenges greater or lesser that do not. For example, in the context of exercise, there's a certain duration and intensity of training that produce good exercise adaptation, good changes in your muscle cells, for example, in your cardiovascular system and your pulmonary system, that will make you better able to exercise under those conditions, use less energy, and so forth, cool your body more efficiently. All kinds of things happen that are good. But if you exercise too much, your body will not be able to make healthy and positive adaptations, and you'll have a condition called overtraining, which is a quantifiable condition in which you will have degradation and loss of performance instead of improvement in performance with adaptation to exercise. Of course, if you exercise too little, nothing's going to happen. If you sit on your couch and wish you were in better shape, it's not going to do any good. So, how do cells adapt to stress? A lot of the changes are as we're going to see, will happen in cells, muscle cells, for example. So how do they adapt to stress? Well, cell signaling and signal transduction. It's kind of confusing because the word signaling pops up all over the place in this discussion. And sometimes we're talking about extracellular signaling, and sometimes we're talking about intracellular signaling. There are both types of signaling, and then there's a crossing over point called transduction. An extracellular signal will be observed by a cell, and that will trigger some changes in the cell. That's called transduction. And then we'll have intracellular signaling. So try to not be put off by the kind of similar terminology used to describe the two domains. All right, so cell signaling, meaning extracellular signaling. Cells communicate with each other using chemical messengers oftentimes. And that's what helps the cells coordinate cellular activities for more complex responses to challenges. They allow us to maintain homeostasis. And the extracellular signaling mechanisms can be classified into a number of different definitions. Intracrine signaling refers to a cell in response to a challenge or a signal coming from outside, producing some changes inside, some signaling pathways inside the cell that will now produce some new cell behaviors. Juxtacrine signaling involves a cell, upon being challenged, releasing a chemical messenger or a signal that will then be transferred into a neighboring cell and change its function, its ability to do what it does. Autocrine signaling, this may seem kind of crazy, but oftentimes cells, when faced with a particular signal or stress or a challenge, will actually release a chemical that binds to a receptor on their own surface. So a cell releases an agent that changes its own behavior. And it's funny that it has to have this sort of loop to an extracellular receptor, but that is actually a fairly common observation about how cells react to various extracellular signals. Paracrine signaling refers to chemical messengers released by cells in response to a challenge that then diffuse to some cells in the nearby region, but not necessarily the adjoining cells. And finally, endocrine signaling, the most distant kind of signaling in which chemical messengers are released into the blood, circulate throughout the body, and then bind to receptors on cells, triggering transduction and changes in the signaling within those cells and the behavior of those cells. All right. So, signal transduction converts signals into cell responses. I've already kind of defined that. So when cells receive an extracellular signal, that signal has to kind of get inside the cell And there's a receptor on the surface that, or sometimes in the cell, that's going to bind to a chemical messenger and then initiate signaling within the cell. That process of converting an extracellular signal to an intracellular signaling pathway is referred to as signal transduction. Transducing means to change energy from one form to another, change that chemical signal exteriorly into some changes inside the cell. What are some of the things that may happen? How will the cell's behavior be changed? Proteins, new proteins may be synthesized. Stress proteins, for example, may be synthesized, including heat shock proteins. That's not the only proteins that may be synthesized, and we'll talk about some specific proteins that are induced within muscle cells in response to certain types of exercise. But anyway, stress proteins, heat shock proteins may be produced in cells in response to temperature challenges, or free radicals, as we'll see. And then heat shock proteins can stabilize or repair damaged proteins within cells. What are some of the stresses that can trigger this process, trigger this production of stress proteins and heat shock proteins? Elevated cell temperature, lower cell energy levels, right? If muscle cells are operating at very high capacity, they consume the energy available, the ATP and phosphocreatine available in the cytoplasm. Abnormal pH. We will talk about, in the next chapter, how the blood pH becomes more acidic during high-intensity exercise. So dropping pH can induce stress protein synthesis. Alterations of cell calcium. Way later on, we'll talk about muscle cells and how they are turned on and activated, and it'll turn out that the ion calcium entering the cytoplasm triggers muscle contraction, but it also triggers changes in the cells that will allow them to adapt to the exercise they're being challenged to do. Pretty cool. And also, finally, free radicals. During exercise, oxygen-using cells will produce free radicals, and the more intense the exercise, the more they'll produce. Free radicals are very reactive chemicals produced from oxygen that will then bind to and alter the chemistry of proteins and DNA, and it's a destructive process. But it actually turns out it activates hormesis in the right range. It's destructive, but yet it causes the cells to adapt and change and be able to better respond to the challenges of exercise and the conditions of exercise. Pretty cool. So how do new proteins get induced in cells? We just said that these challenges will lead to signaling pathway activation, and new proteins, stress proteins, are going to be produced. Well, the chromosomes in your nucleus of your cells, they all contain the same 46 chromosomes. They're made up of many genes, thousands of genes. Each gene, in turn, is a recipe, essentially. It contains the sequence information for making a specific protein. Proteins have specific sequences of amino acids, and the information for that sequence is in the genes. Genes also have a region that allows them to be turned on and off, so we can regulate the genes. We can decide when to activate a protein, such as we're talking about right now, activating stress proteins in response to exercise challenges. Transcription factors, or activators, are specific kind of messenger proteins in cells that can bind to the promoter and turn on a gene. What do we mean by turning on a gene? When a transcription factor binds to the promoter, the gene will be transcribed. That means that some machinery will come along and make a copy of the gene in the form of messenger RNA. In fact, make a whole bunch of messenger RNAs, identical RNAs, and those will be touched up a bit and then sent out into the cytoplasm from the nucleus to bind to ribosomes, and ribosomes can read the sequence information in the messenger RNA and then synthesize a protein with the right amino acid sequence. That's called translation. So transcription factors turn on transcription and translation. So, in the context of exercise, we have signaling pathways being turned on. Those in turn activate specific transcription factors, of which there are many in the cell. And then the transcription factors go to specific genes and activate transcription and translation, and we get the stress proteins or the other structural proteins of a muscle cell, whatever it may be. Turns out that resistance and endurance exercise promote different signaling pathways, and then we get different adaptations. Different proteins are activated in response to those two types of exercise, and very different outcomes result. You can even see those very readily by just looking at the cells after some bouts of exercise. And here's a little cartoon of a muscle cell. This is one little section of a skeletal muscle cell truncated at each end. These long cylindrical cells, just recapitulating what we've been talking about. We have muscle contractions during exercise and some changes in the contractions themselves, plus changes in temperature and pH and calcium concentrations and so forth. And all those things activate signaling pathways within the cell, this domino-like turning on of more and more signaling molecules, which then activate transcriptional activators or transcription factors, which go into the nucleus. We have multiple nuclei you see here. Myonuclei just means nuclei of muscle cells. Goes into the nucleus and initiates transcription of genes in the DNA into messenger RNA, which will then go out into the cytoplasm, bind to a ribosome, and be read or translated into protein. So that's just a cartoon representation of the same process. Stresses to the cell during exercise lead to changes in protein production that will change the properties of the cells. They will adapt to those challenges. This is just something I pulled out of a research review article talking about hormesis. It's a pretty interesting sort of study area in molecular biology and biological science. And here's a bunch of different types of stresses that can lead to hormesis, adaptations, improvements in the body's ability to respond to different challenges. And in response specifically to exercise, here's some of the things that happen. We have free radicals being produced. We have calcium ions coming into the cells, into the muscle cell cytoplasm. We have energy being consumed, and that's going to activate transcription factors. And here's a list of some of the actual names of the transcription factors that may be turned on. And what are we going to synthesize new in response to that? Antioxidants. Mop up those free radicals so that they don't damage the cell. Protein, heat shock proteins. Sometimes they're called chaperones. That's just another name for heat shock proteins and stress proteins. Growth factors. We'll also have growth factors coming in. For example, we didn't talk about that, but it's just a cool thing that will happen. Growth factors go out from the muscle cells and trigger the sprouting of new blood capillaries after certain types of exercise so that more blood flow can come to that muscle cell. If it was starving for oxygen during the exercise bout, it will induce the growth of more blood vessels into the muscles, and we can actually measure that. We can actually take samples right out of someone's muscle and make a micrographic thin section and stain it and count the capillaries and see what's happening. Anyway, that's our story with respect to homeostasis, steady state, and as well as adaptation, acclimation to challenges, whether environmental or just in terms of the exercise challenges we demand of our muscles.